very scarce allocated resources to other groups. So it's very important that we're out there on raising awareness for the disorder on Capitol Hill. And so that's what we've been working on, but it's very, very important. That's why we're signing letters to your congressman. So we're going to get virtual meetings going so they understand Gaucher disease and what our legislative agenda is, from newborn screening to home infusion benefit in Medicare Part B. So how I got meeting this community is through the idea of Medicare home infusion. Medicare, when it was established, there wasn't home infusion for anything. It was basically health insurance for elderly over 65 Americans. That was one of the largest populations of poverty in the United States at the time. The idea, obviously, with Medicare started with Franklin and Eleanor Roosevelt. It did not get passed till LBJ. But my point is, literally speaking, it's still a little archaic. It hasn't really been updated. There was the Medicare Modernization Act 20 years ago, which included basically a new prescription drug benefit, but it did not update like a home infusion. Again, there weren't that many products to be infused. Plus, there hadn't been a global pandemic at that point. So, literally speaking, one group went and got a home infusion benefit, and that was the primary immune deficiency community. But really, what COVID has taught us is that everybody should have the opportunity on Medicare to obtain their treatment if they're being infused with treatment in the home setting. First and foremost, it's less expensive. Why I say that is, if usually in commercial insurance, home infusion is the standard site of care. So unless the patient wants to opt to go to a treatment center or an infusion clinic. So it's very important that our members of Congress know that. They're really starting to understand that, especially after COVID, that people shouldn't expose themselves to a potentially fatal virus to go in and infuse with the treatment they need to help live a normal life. So where are we? We've met with an office. We're drafting the legislation. It's going to be generally for lysosomal storage disorders, more broadly, not just Gaucher disease. We're going to be we're forming a coalition with partner groups, Fabry, Pompe, MPS, to try to put together a coalition to move this forward. We're going to need each and every one of you. Now, you might say to me, well, no, I'm not getting, I'm not on Medicare, or I'm taking the oral therapy. It doesn't matter. You never know what's going to happen. You might be 5 years, 1 year, 2 years away from Medicare. So it's important that we put this in place so that when you hit 65, or if you become disabled, you have access. That's how you obtain Medicare benefits in the United States. There's 2 ways of doing it. Well, there's actually 3 ways of doing it. The first way is to turn 65 years old. The second way is to get disability. That's a very difficult process, and that's probably a story for a different day. The third way is to have end-stage renal disease. And the fourth way is to have ALS. I don't think anybody wants to go that route. So it's very interesting that we make the most vulnerable of our society, which is the elderly and those with disability, actually not be able to get home infusion. It's very counterintuitive. So we're building this patient advocacy coalition. Everybody in this room has to be involved. That's why I've been asking you who your congressman is. I had a great talk with my friends from Phoenix. Actually, a congressman who I worked with many years ago went on a double date with some of the family here. We had a great story about that the other day. I wanted to get my congressman to sign on to the bill, the Ricky Ray bill. So I called his office, and I said, I'm Jim Romano, I live in the district, and I want a meeting with John Fox. And so they said, okay, sir, we'll give it to you on a Friday. And I was a freshman in college, but I was home for the summer. And it was the same day as my girlfriend's prom. And it was at 4 o'clock. So, not a big deal. Okay. So I show up at Congressman Fox's office in my tuxedo. And he proceeded to be an hour late. And they kept saying to me, well, we could reschedule. Nope, we're going to get this done. So he finally comes in, and the first thing he said, is this a joke? And I said, no, I'm on the way to the Archbishop Carroll prom with my girlfriend, but I need you to co-sponsor this bill. I showed pictures of my uncles, and he said, oh, absolutely, I'll co-sponsor. He goes, not only did he say co-sponsor my bill, he goes, I'll get your bill through Congress, which he was a freshman member of Congress. It's really hard for them to do that. But my point is, years later when the bill passed, he spoke at the press conference afterwards, and he talked about me wearing my tuxedo. So you have to do something to make you memorable. It's the important thing. But my point really in saying all this is it's very important that we get your story out to everybody. They have to know Gaucher disease. They have to know what you go through, diagnosis, what your treatment is. So I'm looking forward to working with you all on putting together our advocacy coalition. Newborn screening. We've heard about it this morning in the presentations. We're working to get Gaucher disease on on newborn screening in different states. I know that we're going to be working on trying to get it on RUSP eventually. I'm actually very happy to say that Aviva and I had a call with a state representative from Massachusetts, and he's offering to introduce a newborn screening bill for lysosomal storage disorders, including Gaucher, in Massachusetts. And so I'm glad to make the announcement. He's going to be dropping the bill, and we're really excited. So hopefully I saw there's some Massachusetts people here. We'd love to work with you. This is going to be a big deal. We're going to have to build a coalition, but we're going to go state to state and get Gaucher disease put on the newborn screening in those states. So we're very excited. I'm glad that we've gotten move it this far. So I'm very happy. So we'll be working in different states in the future, and I'm glad to bring my expertise. I like to work with legislators, so I'll learn the issues. But I'm one of these guys who I work with the legislators in different states. So I'm hoping to use my vast way of contacts to help this community and to work with you. So I'm very excited about this. So obviously, I'm really excited about Massachusetts. Okay, so what's going on in Washington? Right now, Congress is on a break. It's on a break to the election. So I don't care how you vote, just make sure to go out and vote. It's important that you do that. Vote your conscience any way you want, but I just have to say that. So what's going to happen on November 8th? All 435 members of Congress, the members of the House of Representatives, are up for re-election. So next year's House is going to be very different. There's also 35 Senate seats up this time, 39 state and territorial governors, and over 6,000 state legislators. And it's all very important that they know what Gaucher disease is, but we'll get to that. Major changes are coming. This Congress has been really kind of odd because they've had a very high number of deaths and resignations and a high number of retirements. So, so we have a very new Congress when we show up in January. So it's going to be very important to get everybody involved. But that's also a great opportunity. You want to get with people when they're new, because after a couple of terms, okay, they've been there a while. It's sometimes a little harder to get them interested in issues. So if we get them interested, if we know, if they know what working with the community now, it's going to be very important. So we're in a really great place. The Senate, again, same thing. There's a lot of retirements. One senator just resigned. So there's going to be a lot going on. I want to say this because it's actually very important. So there's two ways of really getting what you want out of Capitol Hill. Actually, really three ways. The first is you pass a bill. We all remember Schoolhouse Rock. I'm a bill on Capitol Hill. Yeah. So how a bill becomes law. It passes the House, it passes the Senate, and then the president signs it. And believe me, that could take decades. The second way is utilizing the appropriations process. It takes bills years to pass. I mean, really, honestly, unless it's emergency legislation or something. However, every year, Congress has to pass the budget, and they pass a budget resolution, and then they have what they call it, the 13 separate appropriations spending bills, where they actually fund the whole government. The second biggest, the largest one, is the defense, the defense appropriations. I mean, that's the biggest. The second biggest is the Labor, Health and Human Services, and Education appropriations bill. With every appropriations bill, there's an accompanying report. And all that report language has the force of law. So it's working to see how we can utilize, the appropriations process to promote Gaucher disease and to promote our issues. So that's something that we'll be working on. But why I'm saying this is if you're a member of Congress as appropriator, we're going to be coming to you. But the chairman and the ranking member on the Senate Appropriations Committee are both retiring. So you're going to have a whole new cast of characters. It's going to be very interesting come the next year. Okay, so 2022 saw some major changes in the Medicare Part D program. Medicare Part D is the prescription drug benefit. It's 20, I can't still believe it's 20 years old. I was a young lobbyist working on Capitol Hill, working when the Medicare Modernization Act passed. So I can't just believe it's 20 years ago. The real focus of the Inflation Reduction Act, which you've been hearing about, has been really on global climate change and tax policy. However, there was a large component for healthcare, and it's very important to this community. For years and years and years, when they passed Medicare Part D, there was a gentleman who ran the committee. His name was Billy Tozan. He was a bourbon-drinking congressman from Louisiana, having known him a couple of times. As soon as he gets the Medicare Modernization Act passed, he resigns from Congress to become the head of pharma. One of the provisions in the Medicare Modernization Act was that CMS could not negotiate drug prices. Okay. And then he turns around and goes to pharma, and then fights when the Democrats have been trying to change that for many years. Beginning in 2025, this bill allows CMS to negotiate drug prices. Now, the devil is in the details. Basically, this is phased in over a number of years, and you're not going to really see, I think, a reduction in drug prices because of this quickly. And so, it was a major change. Although pharma is thinking of filing a lawsuit against this. The important thing for this community is, beginning in 2025, Medicare Part D will have a $2,000 out-of-pocket cap. Yeah, you have a 2,000. So you'll still pay in the different tiers. So the way the Medicare Part D program goes is pay your premium, then you pay a deductible, then you pay a co-insurance, and then you move into what was the donut hole. You're still going to have to pay your deductible, whatever that yearly deductible is going to be. But once you hit $2,000, that's it. You're not going to pay 5% afterwards. You hit $2,000, and that's it. That's a massive change. Again, it's not coming for a couple of years. So, but I think it's important that this is a big deal for these communities. So if you're on Medicare Part D, big deal. Medicare, if you're on Medicare, you will no longer have to pay any kind of copay for vaccines, which I didn't realize until it's passed that if you're on Medicare, you actually had to pay. In Obamacare, you don't have to do that. And now for Medicare patients, you'll be in good shape. And then in the private insurance market for Obamacare, you have enhanced subsidies. So during COVID, when the Biden administration took office, in one of their COVID relief packages, they did enhanced subsidies. So if you are on Obamacare and you're having a silver plan, you can get some premium assistance and copay subsidies. They were enhanced, during the public health emergency. This is going to be extended through 2025. So if you're on Obamacare and you're having that, you can extend it into the future. So this is pretty important changes this year that the Congress has made that will definitely benefit. Drug pricing negotiation, the devil's in the details, and it's really far in the future. So I can't guarantee that's going to reduce drug prices. But what I can say is your out-of-pockets are going to go down if you're on Medicare Part D, which is a big deal. As I said, if you're not at the table, you're on the menu. So I think it's real important that I'm so glad to be working with this community. I think this is going to be we're going to have a great time, and we're going to do great things together. Why should you get involved? All aspects of your health care, all of them, are regulated by the federal or state governments. So having you involved with, in advocacy, meeting with your members of Congress, getting to know your state reps, because you never know when that's going to become important. If there's Medicaid issues, because that's a state issue, or accumulators. So one of these wonderful insurance tactics is called the accumulator. How many people have heard of the accumulator adjustment programs? Okay, so for those who don't know, if you're in commercial insurance and you are getting a copay card or some kind of copayment card from your manufacturer to help pay for your out-of-pocket expenses, certain insurance companies will not count that copay card amount towards your out-of-pocket expenses. So you'll never get to your out-of-pocket limit maximum. And so you can still use the card and you're still paying for it, but they're not counting it towards it, and it's making your other medical expenses more expensive because you're not hitting that out-of-pocket cap. Some people will hit it the first time when they fill their prescriptions. So insurance is regulated two ways. Because there's an old bill from the 1940s called the McCarran-Ferguson Act, which basically allows your state to regulate your insurance, most insurance, unless you're in what is called a self-insured plan, and your self-insured plans are regulated through the Department of Labor. Okay, so just giving you quickly to make sure you understand that. It's very important when there's like these accumulator, there's coalitions in states to change state law to ban this practice. We hope to get you involved. We're involved in the Pennsylvania one. There's a couple of states that GCA is involved with, so hopefully we'll get you involved with that. So it's very important. I mean, compliance, health insurance coverage, health equity, privacy, your voice is very important. We're gonna have new opportunities. We're gonna be working with you, as I said. I'm going to look up your congressman. We're going to set up a virtual meeting, and then we're going to talk to them about home infusion. I can't wait to work with you guys on this. Fantastic. A good friend of mine, Kim Bernstein, who is one of my mentors, said, all you need to become a great advocate is a big mouth, a clear message, and no shame. Here's my telephone number or my email address. Please call me, email me if you have any questions. I'm at your service. I love working with you and talking with patients, so looking forward to working with you. Does anybody have any questions?
Time for a few questions.
Yeah. On the Medicare home infusion.
Yes.
Is there a reason why you said you're going together with the other lysosomal storage
That is a great question, and that is something that has been in my mind for a while. Here's the problem. I hate to say, here's the challenge with that, is that a lot of the other patient groups want to do it there, they want to just have it for their disorder. Aviva and I had talked, and we said, let's try to expand it. I would love to see a rare disease home infusion bill. I think most members of Congress would shy away from that because of what they perceive to be a large price tag, even though so unfortunately it's going to be piecemeal until somebody wakes up and says,
Wider representation, but also larger cost.
yes. Now, it's not going to be a cost, but
The cost is cheaper to home infuse.
yes.
That's an educational challenge.
Yes. Yes, that is the educational challenge to me. But the important thing is we didn't want to do it that way. We wanted to build a bigger coalition so that when somebody says to us, well, why are we doing this? Like, we could say, well, actually, we're doing it for our section of the community. Yes, ma'am.
Can you give us a ballpark estimate of how much cheaper it is to home infuse, and can't you propose it that CBO is likely to cost it as a negative, and that, this would be great for Congress?
Don't have the claims data yet, but we're going to work on that to find. But here's the problem with CBO. What you're talking about is called dynamic scoring. They don't do dynamic scoring because they don't look at what the potential cost savings is for any program. They only look at what potentially could cost. So that's the problem. That's why we would have to do it with our claims data, looking at the administration. It really just comes down to the administration on how much the nursing will be, because the treatment will be the same whether you go into the, so it all comes down to just figuring out from looking at claims for people who are in Medicare and going to the infusion clinic now versus those who are in private insurance, looking at who's receiving it. So that's something that you might get an email saying if you would like to share your EOB on your administration, to black out your name, but send that out highlighting your EOB. So it's very important that if you hear from us and say, everybody knows what your EOB is, right? Okay. For those who don't, it's your explanation of benefits. You usually get a statement from Blue Cross or from whoever your insurance provider when you, with basically, and I saw this with another disease group, these huge numbers, and then this is how much we're paying, and this is how much you have to pay. And so once we see their EOB, we could say, okay, this is how much Medicare is paying, this is how much private insurance is paying, and then we can go from an anecdotal evidence, reason and get to that. So yes, that's what we're going to be working on. Oh, sorry.
Republicans have vowed to eliminate the negotiation of drug prices and the 2,000 cap if they.
Okay, so they do say that. So there's, in the election cycle, the party out of power who is running has stated that they're going to repeal those aspects. Well, they want to repeal the whole Inflation Reduction Act, and there is going to be voting on that. Importantly speaking, number one is right now they don't have the White House. So if the Republicans do take back Congress, and they do pass that, it probably there'll be a vote in the House. There won't be a vote in the Senate because, well, there could be a vote in the Senate, but the president will veto it. There's not the 60 votes or the 67 votes to override a presidential veto in the Senate. So, you don't have the 2/3 in the House either. So, at that point, so that's an important aspect. They're going to try, but probably won't succeed. Now, if they had unified government, they might be able to do it. But as of right now, it's there till 2025. Go ahead. Sorry.
Based on this question, so let's say that they have the majority and it passed. Can you tell us how is that going to affect us?
So if if the Republicans, this is all hypothetical because the election hasn't happened yet, if the Republicans were to take back the House and the Senate, they could set up they could have, like they did this with Obamacare, they were going to repeal it. So they voted, every couple of months they brought up a repeal, that they were going to repeal the bill and then send it, and they voted 100 times to do that. Because they couldn't get the other body to do it, it just died. What I'm saying is, for at least for the remainder of the Biden administration, is even if they were able to pass, the president would veto it. So as of right now, it's pretty safe. Now, in a couple of years, if there's a Republican president, Republican Congress, then they could possibly repeal it. However, as we saw with Obamacare, everybody got motivated, they called their congressmen, they called their senators, and they were able to prevent that. It's very hard to take away something when people start to like it, and most senior citizens are going to be very happy with and rare disease patients with a $2,000 cap on their out-of-pocket expenses. So there's going to be politics involved in that. I wouldn't worry about that right now. And then we have to get motivated and contact our members of Congress again and let people know how important this is to this community. So thank you very much.
Thank you. And I'm sorry we didn't get to all the questions. Jim is going to be available during our break in a little bit, again, at the booths. So please, if we didn't get to your question, I'm very sorry, but definitely go find him during that time, and he wants to answer all your questions. So now is
Yeah, take your tickets out. And everybody promise right now you have no more than 10. Promise? Yes? Double promise? Okay, so the first is the number 2 prize, which is the Apple EarPods. And I'm going to...
we have to be present in the room.
What? Oh, and you have to be present in the room, so...
I'll set a 60-second
Don't
timer.
leave. Oh, she left. Here, my eyesight. Oh, here. My eyesight these days. All right, so the number is.
Wait.
Oh, they went to get him. Got it. Okay.
All right, bring in the tickets. Come on in. Get your tickets.
Oh, you're bummed. I hope you don't win. Okay, so the number is 3, 5. Are we okay? All right, starting again. 3, 5, 1, 6, 2, 5. No! Woohoo! Are you going to give those to your husband? Do you want to choose the second one?
All right, grand prize. I'm a little nervous.
All right, you guys. My heart's beating. 3, 5, 1, 4, 6, 1. Four, 6, 1. No. Bobby! Woo! All right, so who do we have next? What time is it? 1 o'clock. There it is. We're right on time. Our next presentation is by AvroBio, and we'll let you do your own introductions.
Hello. Can everyone hear me? Okay. Better? Okay, great. Maybe just a little bit higher. So I want to thank everybody here for giving me the opportunity, especially the Gaucher Community Alliance, for speaking about our current and ongoing clinical trial in Gaucher type 1. So just to let you know that there is an active study going on that you can find on clinicaltrials.gov, and it's entitled, and just bear with me because it's a long title for me. So it's called the GUARD-1 Trial, an open-label, multinational phase 1-2 study of the safety and efficacy of ex vivo lentiviral vector-mediated gene therapy, AVR-RDO2, for participants with Gaucher type 1. So, yeah, exactly. It is quite a mouthful, so we typically just call it the GUARD-1 study. So if you hear me say GUARD-1, that is what it is. So, but I do kind of want to unpack a little bit about the title. We did say it was an open label, so what does that mean? That means that the patients, the investigators, the clinical trial physicians, as well as the sponsor, AvroBio, knows what the treatment is. We're not comparing this treatment with ERT or SRT. We are doing it as a single arm type of trial. Also, we're talking about vector-mediated lentiviral ex vivo. And what does that mean? It means that we're using the patient's own stem cells and inserting an enzyme in there, and then re-infusing that enzyme into the patient's body. So we're not having like healthy volunteer donors or anything using their stem cells. It's the patient's own stem cells. So I am obligated to give you some disclaimers here. So AVR-RDO2 is the investigational gene therapy product. It is investigational, experimental. And we are developing it for individuals with Gaucher disease. It has not been approved yet by the FDA or any other regulatory agency. And if anybody's interested in treatment options for Gaucher, including this trial, please speak to your healthcare provider. I am an employee of AvroBio, which is located in Cambridge, Massachusetts, and I am joined by my colleague Elise Thomas in the back as well. And my role at AvroBio is to oversee and manage all the clinical trials at AvroBio, so not just with Gaucher, but we have a few other programs in other indications. And we once again thank the Gaucher Community Alliance. I'm trying to talk slower than I normally would because I am from Massachusetts, so I have a little bit of a speed problem, so bear with me here. All right, so GUARD-1. As I mentioned, it is for Gaucher type 1 patients. We are looking at the safety and effectiveness of the AVR-RDO2 product. And if you're interested in this trial, I encourage you to speak with your healthcare provider, as well as look at clinicaltrials.gov for more of the eligibility information, as well as we do have a website here. Oops, just kidding. Let's see if I can get this right. Basically, the AvroBioGaucherTrial.com. It'll give you some more information about the trial on there as well. It's very consistent with what's on clinicaltrials.gov. If you go there, you'll also be able to fill out a very short pre-screen questionnaire and then be assigned to a case manager who will review the information you completed on the questionnaire. And then what they'll do is speak with someone on AVROBio to see if you can be assigned to a clinical site. And there you will be able to speak to a physician to really understand the risks, benefits of the trial, as well as, hear about how other patients have fared on the trial. If you are interested in this, you have to have a confirmation of diagnosis of Gaucher type 1, be between the ages of 8 to 50 years of old, and have been at least on enzyme therapy, or if you are on SRT, there are some stipulations there where you would have to come off of SRT for at least 12 months prior to enrollment in the trial. But on ERT, you can remain on ERT, and then a week before, or 10 to 12 days before receiving the gene therapy infusion, you would come off of ERT. I will entertain questions if anyone has questions during my presentation. I won't wait till the end, so feel free to let me know if you have any questions. Okay. All right, so as I mentioned, we are looking at safety and looking also as how well participants have tolerated the AVR-RD02 gene therapy product. We are looking at the effects of the product on markers, biomarkers that I know many of us have heard about throughout the last 2 days, as well as how are patients feeling, the quality of life. This is really important here at AVROBio. Every meeting that I attend internally, the first thing is, how is the patient going to feel? We are a very patient-focused company, so it's patient first. Every meeting, doesn't matter if it's the scientific, if it's the legal, the finance, even all the way up to our executive team and our board of directors. We are really focused on the patients. So it's really important to see not only how the product is doing from a scientific and medical, but it's also to see how the patient is feeling, because that's really important. So I kind of want to share kind of like this overall high-level study design of our clinical trial. So if you're interested in the trial, as I mentioned, if you go to that website, you'll complete a pre-screen questionnaire, and then we'll be assigned to a case manager. And if you do get assigned to a clinical site and you speak to the physician and their staff about the trial and decide you want to participate, you'll sign an informed consent form, and then you will undergo a few different assessments, lab assessments, blood draws, and confirmation of Gaucher disease. And this could take up to 30 to 60 days. Once the physician receives all the results, he or she will communicate with AVROBio to confirm eligibility. If you are eligible, then you will enter the next phase, which is the baseline visit. Here you'll have some blood draws again, just to assess and to be able to use as comparison to other time points after receiving the gene therapy assessment. Once you complete the baseline visit, you'll go into a state of mobilization. So mobilization is a time where the patients will help the bone marrow release the stem cells into your bloodstream. And that takes about approximately 4 days. And then what happens is, then you have the apheresis section, or days, which is 2 days. That is in itself taking the actual stem cells and sending it out to get it manufactured to make our drug product. So once again, as I mentioned, our product is one where we use the patient's own cells, so you have to extrapolate or extract the patient's cells, and then we go and we send it to a lab to actually put the gene into one's cells here. Then it comes back to the site, and then the patient will undergo a conditioning period. And the conditioning period is about 5 to 6 days. And in that stage, the patient's bone marrow is made space to receive the infusion, the gene therapy infusion. And then for 1 day, you'll actually.
Made space? You said those 6 days made space. What does that mean?
Sorry, say that
You
one.
said it's made space.
Yeah, so the conditioning helps clear up space so that you can accept your own stem cells back again.
What's done during the conditioning?
The conditioning, yeah, so you're given a product called busulfan to help clear out the space in the marrow.
What type of drug is that, sorry?
Sorry?
What class of drug is that? Like, what is busulfan?
Busulfan is a conditioning agent. It's normally used in oncology.
Like a chemotherapy?
It is a chemotherapeutic. But it's in a lower dose than what you would get like if you were actually, an oncology patient.
But will it still have like similar side effects for the patients?
There are side effects along the lines of busulfan, yes. Did I answer your question, sir? Yeah. Okay. All right, so then what happens is after they've cleared up the space in your bone marrow, then you're eligible to have the actual gene therapy. It's a one-time infusion. And then after the gene therapy infusion, you're followed up for at least a year at different intervals just to see how your body's doing from the gene therapy infusion, as well as looking at your Gaucher disease. And then also looking at your quality of life. Then because this is a gene therapy trial, we are obligated to follow a patient for up to 15 years post-gene therapy infusion. So what happens is participants have the ability or the option to enroll into a follow-up study, a long-term follow-up study for the remaining 14 years. So the first 4 years, you would come back to the site every 6 months, and then the remaining years once a year. Yes?
Is the conditioning process just a part of the trial, or is this designed to be part of the actual gene therapy treatment?
It is part of the trial, but it is in order to receive the gene therapy. So it would be part of, if it was to ever be approved, it would be part of the.
So for someone, let's say it's approved by the FDA and I want to get the gene therapy, so I may eventually, if I want to do that, face, like the potential chemotherapy side effects. And it's not just part of the trial, but it's part of the deal.
It's part of the deal, yes. I know that was a really short presentation, but I really wanted to have time to answer questions here for any of you. We also have some informational sheets available in the exhibit halls, and I'm happy to answer any questions on a one-on-one basis, after this presentation. Yes.
How many people have enrolled in the study so far? How many people have completed the gene therapy?
Yeah, so to date, we have 2 that have been dosed with the gene therapy, and they have been followed for over a year.
Can you share any of those results, or that's not, can't do that because it's only?
Yeah, at this time I cannot share the results, but in the next few months before the end of the year, AVROBIO will be sharing more of the program as well as the results to the community, and we will be definitely mentioning this with the Gaucher Community Alliance so that all of you can be aware of our results. Oh, yes, sorry.
Can you share the site locations for where the trial is happening?
So currently they're in the US, and we will be opening another site in Canada. So we have a site in Hackensack, New Jersey, Pittsburgh, Pennsylvania, and Iowa. I can't remember the city in Iowa, but it's Iowa. And we are, actually, we just also opened a site in California.
Do you know northern or southern?
UCSD, so Southern. Yes.
Can I ask a question about your inclusion criteria? It said 24 months ERT or nothing in the last 12 months, so is it 2 different arms?
Yeah, there are two different kind of patient populations that we're looking at. So if you're on ERT, at least on a stable dose, you have the potential to be eligible. I can't confirm you're eligible, right? But if you have not received any therapy, or if you've discontinued therapy in the last 12 months, so either SRT, you're considered like a treatment naive for the purposes of the trial. Yes?
If you're on SRT, could you switch over and for how long? Switch over to ERT?
You have to be on ERT for at least, I think, was it a year or something? Yeah, yeah.
And you're too old if you're over 50.
I wouldn't say you're too old, you just don't meet the eligibility. I'm not walking down that road. Yes.
During the trial, and I guess right before the treatment and during the preparation phase, patients are relatively sick. I mean, how are they feeling in terms of keeping down a job, and what are their lives like during the trial?
Yeah, so we do have patients that are participants that, you know, in all of our clinical trials, because we're a lentiviral gene therapy company, but specifically for Gaucher, we work with the patient, the sites work with the patients to really accommodate that, as well as we allow, you know, a patient's caregiver or family member or friend to accompany them as well during this time.
Just to be a little bit more specific, like especially during the time during the conditioning and after the conditioning, like what that recovery period is like and how much time one would be expected to take away from their job or their family if you're a parent or if you're working full time.
Yeah, so the conditioning is about, so the conditioning period, although you're not getting conditioning every single one of those days, but the preparation of it is about 10 to 11 days. Then it's a one-time gene therapy infusion. And then you are followed for at least 7 days right after that. So you are considered an inpatient during this time. A lot of the other procedures are considered, you know, outpatient.
That's 17 days.
It's approximately 17 days, 17 to 20 days.
Do hospital beds have internet?
Any other questions?
Thank you so much.
Thank you. Oh. I'm sorry.
They will have to be in those locations that you mentioned, right? They can't be anywhere else?
Currently, those are the sites that we have open, but we do offer assistance for patients and their caregivers to travel. And so the sites can provide more information on that.
Thank you.
Thank you.
Our next presentation in the next 2 minutes is Prevail Therapeutics. Do you want to frame these winning tickets? Actually, I'll do it this way. All right. I hope I get as many exciting, interesting questions. So, Sarah Newhouse, I'm one of the senior medical directors at Prevail Therapeutics. I'm honored to be here at the inaugural meeting and have the pleasure of interacting with everyone today. Primary focus of the talk today will be our GD1 trial called Proceed, but wanted to start with a little bit of background on the company and what we're working on, and then it's actually great to be staggered behind AvroBio because I think we were probably getting similar questions at our booths. Some of the most common questions are sort of, what is gene therapy? How are they similar? How are they different? What can we anticipate? And I think to the degree we can be candid with you about the differences and the pros and cons, we should do that. And just to let you know that your questions really help us inform making our trials better and what we need to consider. So we are huge advocates to hear all of your honest feedback and what you'd be willing to do and participate, because if you're not, then we can't really move forward. So your feedback is really vital. So thank you for your time and attention. The same medical disclaimer, these are experimental, these are not approved. So this is not intended to be medical advice. Please speak with your physician. We're going to do a brief introduction, and by we, I mean I am going to do a brief introduction on Prevail. It won't be a quiz just yet. Review just gene therapy in toto, in general. What is it? What are the mechanisms? How is it working? How do we think it works? What are our Prevail's AAV-based gene therapy approaches? What are we working on? And then more specifically, an overview of the trial that is pending to start. So that'll be the most exciting element. In total, Prevail Therapeutics, which was founded in 2017, really with the main mission of attacking monogenic, so single gene, neurodegenerative conditions. So rare diseases typically, but from one that we have a good understanding of a gene to protein to disease relationship. And so really targeting those specific mechanisms. The pipeline, kind of what we're working on right now, we actually have 3 ongoing trials with one common product. So there are 3 different manifestations, as you guys know very well, of GBA1 spectrum disorders, from Parkinson's to type 1 to type 2. And so there are 3 ongoing studies, I should say 2 ongoing and 1 pending, using formally the easy to say PRO01. And I guess I should mention in 2020, Prevail was acquired by Eli Lilly, so we got blessed with this lovely LY3884961. We sometimes casually, colloquially still say PRO01 for ease, but those are the same compound, essentially the GBA1 AAV vector. We also have a trial ongoing in a different condition, frontotemporal dementia, with progranulin mutations, so different. So the focus of the talk today will be the elements in this sort of purplish blue color. So what is gene therapy? So this is just a step back, not just our vector, but what are we doing in general? So the goal is essentially to slow or to stop the progression of a genetic disease. I very cautiously use the word cure because I think we are always humbled by what we're able to do, and I think it's our job to hope for that. But at this moment in time, our goal is really to address conditions where they are and stop the progression, with hope ameliorate future disease worsening. But that is the main goal. I think a lot of what we're struggling with right now as patients, as families, as physicians, and as drug developers is that regulators really require the patient to have symptoms before we can treat you, because we need to know that you have a physical manifestation of your disease. And what we heard from all of our medical experts is the really key time to treat you is before you're ill, is pre-symptomatic. And that is the goal. And I say that drug development is phase-wise, it's iterative, but the very first phase, for better or worse, is almost always in adults, if possible, and almost always in patients who have symptoms. And that's just the sort of painful drug development process we have to go through. And the goal would be, with success and with evidence and with safety, we move forward to younger, less affected, and even pre-symptomatic patients. So I think we are all marching towards that goal. We just have some challenging steps to get there. So how is this done? We want to slow or stop. We deliver essentially a healthy copy of a gene, what I kind of liken to essentially replacing a tire. If you run over a nail and your tire is flat and you pull out your donut, you still have the tire that was injured in your vehicle somewhere if you didn't leave it on the side of the road. So you have the mutated gene in your body, but we are essentially transferring a healthy copy of that gene into your cells, which is able to then produce the functional protein and do its job. This is just generally speaking how gene transfer, vector-based gene transfer works. If you have a single gene with a misspelling, we replace it with one that's properly spelled. It makes the protein that works. It gets into all of your cells, using sort of the viral vector's natural ability to do what it does in viral settings like COVID or influenza. Viruses invade cells, take over their DNA and produce what we want them to. So that's essentially the overarching purpose of that. It has been well studied for decades, since the late 80s, early 90s, initially in hemophilia and some other metabolic conditions that made the news. But just to mention, I think 2 FDA and I guess 3 FDA-approved therapies, 2 with AAV that have been most common in the press, are the spinal muscular atrophy SMA condition, so Zolgensma as an approved therapy. And then there's another AAV vector-based therapy, Luxturna, which is for a visual sort of condition. So they had been studied for a very long time. Just to mention sort of back-to-back, what are different types of gene therapy? And these are some of the questions you guys earned tickets with, although I don't know if any of my tickets won any of those, but excellent questions. So Gene therapy versus gene editing. I made that analogy of the tire because gene therapy is essentially the transferring of a functional copy into your cells, but we are not editing your genome. And so that means if you go on to have children, your children will not carry the product that you were given. So it does not integrate into the host genome. It isn't carrying that mutation forward into the next generation. So those are sort of two different mechanisms. I think CRISPR-Cas9 is very sexy in the news, often spoken about. That's not what we're doing here. We're not editing your DNA. We're doing that sort of flat tire replacement analogy, if that still works here. And then we have something in vivo and ex vivo. So what does that mean? Are we giving something to you in your body, or are we taking your cells out, altering your cells, and giving them back? So AAV, vector gene therapy, is an IV infusion. So that is in vivo, so we give it to you in your arm, or your foot, or whatever vein you prefer, in that way, just much like an ERT infusion. And then the viral vector that we use. So we talk about historically, long time ago, adenovirus, a cold virus, was initially used in the early, late 90s for gene therapy. This is its sort of very tiny, less pathogenic or non-pathogenic kind of parasitic virus. Adeno-associated sort of attaches onto adenovirus, but it itself is non-pathogenic in human beings. And there are multiple different types of that. And so that has a smaller sort of carrying capacity, is a bit of a smaller vector, but it has been used to carry single genes for the purpose of transfer and replacement, versus the lentivirus, which is something more commonly used in ex vivo, which the previous speaker discussed. And then immune suppression, that's a huge question, right? Not only the therapy you're giving me, what are the other drugs I have to take? How are they gonna make me feel? How long do I need to be on them? What kind of life can I live while I'm on them? And so there are different types of immune suppression regimens, one of them being prophylactic, and one of them, again, conditioning, as the previous speaker alluded to. We use prophylactic immunosuppression, which are not IV and they're not chemotherapeutic agents. We use 2 different oral agents, which we'll get into more detail later. Essentially, the goal is, although this vector is carrying something helpful, your body still identifies it as a virus. So we want to be able to dampen down the immune system just enough that it's able to allow that vector to do its job without creating any kind of immune response, because the doses we're giving are higher than you would get in a normal viral infection if you were to get a cold or a flu, for example, exposure in the environment. So just
What kind of exposure does that give you to other viruses? Risk of exposure?
what kind of risk if you're on immunosuppression?
Right. On this type of suppression, what are you going to get a lot of colds, a lot of whatever, a lot of...
Yeah, do you mind if I defer to the timetable, because I think it would be helpful? These are generally well-tolerated immune suppression agents, and so we recommend regular hand washing, covering, and exclusion, but it's not akin to a very aggressive sort of oncology type regimen. It's more acceptable. People on these drugs are out working, living normal lives, having exposures. Yeah. So just to highlight again, we do transfer, we don't edit. We do in vivo, not ex vivo. We use AAV as our vector, and we do prophylactic immune suppression. So just to say, those are the 4, in all of the trials that we're currently doing, these are the, and I'll pause so you can get some good screenshots. These are the four sort of harmonizing common approaches Prevail does in all of the studies. I will say, in terms of in vivo, for our PROCEED, for the GD1 peripheral manifestation study, it is IV. For the Parkinson's and GD2 programs, those are direct infusions into the central nervous system space. So those are a neurosurgical procedure. They're in this little area behind the base of your skull, and those are direct infusions done usually by a neurosurgeon or somebody trained to do that. But in the GD1 program or peripheral Gaucher program, that is an IV. But they're all direct infusions. Okay, this is taking a little bit of a closer look of what does the vector do once it reaches the cell. And I like this image because I think it may help clarify where the DNA actually ends up. So as viruses do, it's essentially working a bit as an Amazon truck with a package and sort of delivering this package to the proper location. So the sort of the Amazon or unbranded delivery truck that we use is called, this is not a plug, they have enough, is AAV9, and that is the vector sort of shuttle that we use to take that GBA1 copy to the cell. The DNA essentially finds its way to the nucleus where it lives and essentially creates its own little GBA1 or GCase factory. If you see to the sort of, I guess, stage right or your left, the faulty DNA causing disease, that is your genome that is not being touched, it's not being edited, it's not being altered in any way. So it stays what we call episomal in a different piece and creates the protein. All right, so we also have a very long, difficult to say title, but it has some common features that are actually quite common for a lot of rare diseases. So the short name for the study is called PROCEED, and then the long name is Phase 1/2 Gene Therapy Trial for Type 1 patients with peripheral manifestations. If you notice, and I think we discussed this today, people are really bright about how drug development works, and you're Phase 1, which means you have to get to Phase 2 and Phase 3 and Phase 4, and how long is this gonna take? I think our regulators are trying to understand the challenges and the rarity of the disease. We can't always in gene therapy and in rare disease progress through all of those stages as clearly as with other drugs for high blood pressure, diabetes, and as you'll see, a lot of us are combining Phase 1 and 2. So Phase 1 are designed to be safety, and Phase 2 are designed to be dose finding, and we really have to kind of combine those because we need to know, especially in gene therapy where these are one-time drugs, we have to give you an efficacious drug the first time. We don't have the opportunity to do what's called a dose-finding study where we play around, we give you another dose, we raise the dose. The first dose we give there has to be some evidence of biological efficacy for you to take that risk and for you to have participation in the study. So we kind of combine Phase 1, always assessing safety, and Phase 2, assessing which dose is efficacious.
Does that mean they're giving a particularly high dose to make sure they give enough?
Excellent question. We have to start at a high dose that in animal models has shown to be efficacious. Does that mean we use high doses to start? Yes. We use high enough doses that show efficacy within a range, and typically studies have low and high or low, medium, and high doses as we do in ours as well. But even the low dose has to make some type of physiological GCase protein that you would benefit from. Yeah, so peripherally, yeah, absolutely. You're jumping all ahead to, yes, we'll get to them a second time, but yes, the peripheral dose, no, it's excellent. When we dose intracerebrally, like in the brain for GD2 and PD, we calculate based on brain weight in grams. So a typical adult weight, or for pediatric patients, they have different, a 6-month-old isn't the same as a 24-month-old, so we do calculations for pediatrics. For peripheral diseases, they're based on kilogram of your body weight. So we have low, medium, and high, and then they are given to you based on your body weight. You may have a different dose than someone else. Did that answer? Okay. So aim of the study, as we already said, it's to assess the gene therapy, and it's to assess safety and dose finding. The key elements of the study detail. So FDA mandates that any gene therapy study is 5 years long. We have to observe long-term effects. We have to observe durability because this has to last. So the trial in and of itself is 5 years in length, 15 subjects. So who are we looking to recruit? Would be a total of 15 subjects currently between the ages of 18 to 50. I think I've met a lot of very young 50-plus community that have shown incredible enthusiasm for this study, and I will take that feedback home directly, and we will look into that. But I think it's important, we're assessing adult population. We're looking at 8 international study sites right now, so we're trying to be as global as possible. We understand these are long commitments for the study, and travel is a main piece, but we're looking at 8 sites, and this is one-time therapy. So as it stands right now, AAV-based therapies are single dose, one time. And we mentioned that we're targeting peripheral manifestations of disease, and this goes into a little bit of the nitty-gritty of our inclusion criteria, but we understand that there can be maybe some very mild peripheral neurological symptoms, and the typing of that patient may be complicated, so we can talk about that on an individual basis, but the IV infusion is aimed to target those peripheral manifestations in the blood, in the bone, in the liver, and the spleen. And we mentioned the prophylactic immune suppression. So what are some of our key inclusion criteria? So we mentioned the age. We mentioned that you have a clinical and genetic confirmed diagnosis, which usually isn't an issue in the United States, but some other patients in some other countries have more challenges getting confirmed genetic diagnosis. Currently in this phase 1/2 study, in the United States, we are unable to dose naive patients in the gene therapy trial to start, and what that means is that we will be recruiting patients who've already been on an approved therapy in ERT or SRT for 2 years. And have been on a stable and tolerated maximal efficacious dose for them. So for this initial study, we will be targeting those patients who are considered to be either not non-responding. If you're non-responding, that's a different issue, but suboptimally responding. Is there some element of the disease that isn't being fully addressed or fixed by the SRT or ERT that you're on? And what that breaks down to is essentially one of these peripheral elements hasn't been fully addressed or optimally treated per your physician and yourself and your expert. So that comes down to some detailed criteria on what is the size of your spleen, what is the size of your liver, and how are your platelets faring. Those are the really easiest sort of peripheral ways we can measure your response to your current therapy, and would you be considered eligible to participate. The last one is also bone manifestations, which we appreciate is rare, but these bone manifestations are quite broad. Not everybody has a bone crisis or an AVN or something as severe as a pathological fracture, but we do use DEXA for your bone density, and we use MRI of the bone to calculate a bone marrow burden score, a BMB score, and so those are also ways. You don't have to have a traumatic fracture or something quite severe to qualify. There are ways we can figure out the bone density on imaging and see if that's a possible criteria. Yes?
Sorry, does that mean you cannot have ever had a bone manifestation event, or that you haven't had one recently?
Let me clarify. So that actually means we are looking for patients who have had a bone event because ERT didn't prevent the bone event. So if you did have, this sounds morbid, I don't want anyone to have this, but if you have had a bone crisis or have had a fracture or have still persistent osteopenia, that means the ERT isn't helping. You would meet criteria to potentially be rescued by gene therapy. That's correct. Yeah, so not that we want you to have it, but you would be eligible for the study if you had it. Or if your spleen size is still enlarged, your liver size is still enlarged, if those elements haven't been corrected. Basically, symptoms that aren't fixed despite you being on ERT for 2 years. Yeah. Sure. Yes? Okay. So key exclusion criteria, and I got some really amazing questions from some patients and people, and we will get back to you on some answers. But one of the key criteria we mentioned, we'll be using 2 immunosuppressants. I'll get into those. So if you have any history or adverse reaction to those 2 drugs, that's unfortunately an exclusion criteria. If you have significant neurological impairment, and we could discuss what that is more on an individual basis, that could be decided by your investigator, your physician, that would be an exclusion. If you've had a total splenectomy, and the reason for this is we're measuring the size of your spleen as an inclusion criteria and as response to the therapy, so if you've had a complete splenectomy, unfortunately in this phase 1, that would be an exclusion criteria. But I did have some really great questions on partial splenectomies, and I will get back to those patients about inclusion if you've had a partial and ability to participate, because you still have something we can measure. Progressive bone disease, if you are predicted to require any surgical correction. So anyone that is amidst of an orthopedic need, we don't want to interrupt that, and we don't want to complicate that by gene therapy. So if you have a predicted surgery coming up in 6 months, that would be an exclusion criteria. And then if there are any evidence of other diseases, viruses, cancers, anything else that has been identified from a liver and spleen perspective outside of Gaucher disease would be an exclusion. Okay. So what is the design of the study? I think we mentioned a little bit how this works for dose finding. So there's pre-screening or screening visits with eligibility. We do a lot of infectious testing, blood testing, confirm your diagnosis. We do some imaging as well. And then we have what's called dose ascending, but it's sequential. So you're not randomized into a dose. The way this is organized is we have 3 patients at each dose level, and they're filled sequentially. So 3 at a low dose, followed by 3 at a middle dose, followed by 3 at a high dose. So we wouldn't randomize somebody into low, medium, high. And they happen sequentially so that we can look at the biomarker data, the efficacy data, and most importantly, the safety data and the response. So that after each patient that's examined, we're able to assess their status, their labs, how they're doing, their imaging, and then we would make a decision on whether or not to dose escalate. So it's a small study, 3 at each level. Give me a hot moment. Yes, sir, there is, because the dose that we decide to move forward with, it's not always the highest dose. So if you see we have 3, 3, 3 is 9, double check my math, but whichever dose is safe and effective, most safe and most efficacious, we would move forward into that, into a dose expansion cohort. 9 and 6 is 15. That would be the end goal of this study, would be 15. I think there are a few, let me just double check again. I think there's an additional piece I wanted to mention, which is patients are on ERT or SRT when they're enrolled into the study. And with the assistance of that physician, their treating team, sometimes that's the study physician, sometimes it's a different one, we wouldn't begin to wean that therapy, the ERT or SRT, for at least 3 months, because we want to allow the vector time to enter the cells, produce enough GCase protein to essentially replace the ERT that you're on and do its job. So it would be about 3 months after the dosing, into the study, that we would begin to assess those criteria and then consider reducing and weaning off the ERT to see how the gene therapy is holding. And then we mentioned sort of the 5-year follow-up. So I would say the first 12 to 18 months are the most frequent visits, the most common checks. And then the last 3 years are essentially 6-month check-ins that are more spaced out, recognizing it's burdensome to attend a lot of clinic visits. Okay, any questions on the study design? Yes.
How are you testing the efficacy if they're still on the drug, on the ERT?
Yeah, so the goal is to essentially, we're measuring elements of your liver and spleen volume, as well as your blood counts, as well as your bone marrow, bone density and bone marrow burden scores. So we have the ability to identify you as someone who has not optimally responded to ERT. All of those elements, along with blood biomarkers, will be followed throughout the study. So.
What happens after the initial 3 months?
Right, when the ERT or SRT is weaned or withdrawn, those same parameters have to be monitored, and if there are changes in those, meaning that there's a drop in your platelets, for example, or a change in your liver or spleen size, that would essentially mean you would need to be restarted on your ERT. Is the gene therapy not holding up?
So they wouldn't be withheld with the ERT if it looks like they still need it?
Say it one more time, it wouldn't be,
They're weaned off ERT and then now the levels go up, which means maybe they were on the low dose of this therapy, let's say, for
right,
argument's sake.
right.
And therefore, it's not a high enough dose to be efficacious. So they would be able to get that medicine. So
Absolutely.
they're not suffering from the.
Absolutely. If there's any evidence of clinical decline, you would be restarted on a therapy. Absolutely. Yeah, that would not be withheld. And we're following those clinical elements for that reason. If your treating physician feels, you're not doing as well as you were, we need to restart it, then you do. Excellent questions. Just making sure I'm not missing this side of the room. Okay. Oh, Ness? Yeah, please. You sure? Okay, this is a sort of graphical overview of some of the assessments that would be done. So we have the physical exam, which is essentially a physical and a neuro exam, which I'm sure all of you have done many times. We have an MRI, and we do the MRI of your chest and abdomen, MRI of the bones, MRI of the liver and spleen. We have a number of questionnaires on how you're feeling, patient-reported outcomes, activities of daily living. We have a fatigue scale, and essentially ability to see how you're doing, if anything has changed, if it's improved, if it's worsened, and then obviously a number of blood tests for safety measures for your liver and kidney function, and then biomarkers for the GCase. And you'll see, I just will say the columns are not equal to the time. They don't represent. So the first 3 days, I guess, are the biggest and most widest because that's the most intense period of time. The infusions are typically given either in a hospital setting or in an ambulatory clinic setting and observed overnight. And then, to week 1, week 3, month 2, the visits are sort of spaced out throughout the study. But the most sort of acute period or most frequent monitoring for blood values are in the beginning. At the bottom, I just want to draw attention, there were some questions around immunosuppression. So the day prior to dosing, we administer essentially a high-dose steroid called methylprednisolone. If you've heard of prednisone, It's a medication commonly used for many different conditions. People have had it for asthma or for different dermatitis or rash reasons. It's a non-specific immunosuppression that we use as sort of a medium high dose for adults, and that is an oral tablet that you take by mouth for approximately 3 weeks. And so it's not considered a prolonged period of time. 3 weeks is a decent course, but there are some conditions that take it for years, some conditions that take it for many months. So this is a medium to short course of prednisone. The other agent that we use is sirolimus. It's also called rapamycin or Rapamune. This is a specific drug that has been shown to be incredibly helpful in gene therapy to suppress your T cells. So your immune system, I think, as Dr. Mistry showed his beautiful graphics, there are many cells involved in the immune system. And so the goal is that we sort of tamper multiple cell types so that it doesn't respond to this virus that we're giving, this vector that we're giving. And the sirolimus is on for approximately 3 months with a 1-month taper. And that is an oral medication as well. It is very commonly used in kidney and liver transplants, a solid organ transplant. Typically those patients who receive transplants are on these drugs for life. They have certain levels that we measure in the blood. For the goal of this therapy, we're targeting for about half of the trough levels than we would use in a solid organ transplant. So similar drugs, but at lower doses than we target than transplant surgeons target for things like kidney or liver transplant. So it's a one-time infusion, prednisone for 3 weeks, and sirolimus for effectively 3 to 4 months. One-time methylpred infusion is the day before you receive gene therapy, yes.
So they're infusing the body with this gene therapy even before the resistance? Or don't they need to build up the resistance before they infuse?
So the methylpred,
It's a little backwards is what I'm saying.
The-
They need to first
We-
lower the suppression
we-
and then give the shot afterwards.
we do it that way. So you get the methylprednisolone, you get the steroid infusion the day before you get the gene therapy.
Where in this timeline do you get the gene therapy?
There's not an arrow for that. Very good point. There's not a specific arrow for the gene therapy. The IV methylprednisolone is the day prior. This sort of yellow golden bar is just representing lots of activities, so they're not very clear. Correct. So you essentially would go to a hospital, you would get a steroid infusion, and then 24 hours later you would get the gene therapy infusion.
Prednisone and the stroke,
Sirolimus, same day.
but it's continuing then for several.
Correct. But yes, to your point, we give, prior to the gene therapy, you get a dose of immune suppression so that it does exactly what you said. Yes. Okay, so just to review, these are the goals of what we're trying to gain from the study, and I think they cover most of the categories that people are concerned: safety, efficacy, the blood biomarkers, as well as hemoglobin and platelet counts. We're looking at disease biomarkers, elements of GCase and different breakdown products that were discussed earlier today, spleen and liver volume, energy, quality of life, how you're feeling, fatigue, and then bone and lung disease progression elements as well. So in summary, I think we mentioned that eligible patients undergo screening genetic testing. You receive a single dose of this gene therapy agent, PRO01. We include immunosuppressions before the procedure to minimize potential immune responses, and then we follow up for a total of 5 years. And we also have listings, more detail on clinicaltrials.gov as well. And if you are interested, recommend. We're hoping to have sites open imminently and can share more details when that happens.
Where are those sites going to be in the US?
Do you want to say?
Fairfax, Northern Virginia.
Dr. Gaucher Alpen will be hopefully our first site open in the US. Yeah, absolutely. I think that's it. This is just summary of this, but everyone knows. Thank you so much for your time.
Wow, lots of information. Thank you, everyone. We have a break for the next 20 minutes, and we're going to resume at 2:10.